The electrons carried by NADH can be used to produce 3 ATP molecules, and those carried by FADH2 can be used to produce 2 ATP molecules. We turn now to the process by which the energy of electrons carried by these coenzymes is converted to ATP energy. It is a series of reactions called oxidative phosphorylation, which couples the oxidation of NADH and FADH2 to the phosphorylation of ADP to generate ATP. Before we try to understand the mechanism of oxidative phosphorylation, let's first look at the molecules that carry out this complex process. Embedded within the mitochondrial inner membrane are electron transport systems. These are made up of a series of electron carriers, including coenzymes and cytochromes. All these molecules are located within the membrane in an arrangement that allows them to pass electrons from one to the next. This array of electron carriers is called the respiratory electron transport system. As you would expect, in such sequential oxidation-reduction reactions, the electrons lose some energy with each transfer, and some of this energy is used to make ATP. At 3 sites in the electron transport system, protons can be pumped from the mitochondrial matrix to the intermembrane space. These protons contribute to a high-energy proton reservoir. At each of the 3 sites, enough protons are pumped into the proton reservoir to produce 1 ATP molecule. The first site is NADH dehydrogenase. Because electrons from NADH enter the electron transport system by being transferred to NADH dehydrogenase, all 3 sites actively pump protons and 3 ATP molecules are made. FADH2 is a less powerful electron donor. It transfers its electrons to an electron carrier that follows NADH dehydrogenase. As a result, when FADH2 is oxidized, only the second and third sites pump protons and only 2 ATP molecules are made. The last component needed for oxidative phosphorylation is a multiprotein complex called ATP synthase, also called the F0-F1 complex. The F0 portion of the molecule is a channel through which the protons pass. It spans the inner mitochondrial membrane. The F1 part of the molecule is an enzyme that catalyzes the phosphorylation of ADP to produce ATP. How does all this complicated machinery actually function? The NADH carries electrons originally from glucose to the first carrier of the electron transport system, NADH dehydrogenase. There, NADH is oxidized to NAD+, which returns to the site of the citric acid cycle to be reduced again. As the dashed red line shows, the pair of electrons is passed to the next electron carrier and protons are pumped to the intermembrane compartment. The electrons are passed sequentially through the electron transport system, and at 2 additional sites, protons from the matrix are pumped into the intermembrane compartment. With each transfer, the electrons lose some of their potential energy. It is this energy that is used to transport protons across the inner mitochondrial membrane and into the proton reservoir. As mentioned earlier, FADH2 donates its electrons to a carrier of lower energy and fewer protons are pumped into the reservoir. Finally, the electrons arrive at the last carrier. They now have too little energy to accomplish any more work, but they must be donated to some final electron acceptor so that the electron transport system can continue to function. In aerobic organisms, the terminal electron acceptor is molecular oxygen, and the product is water. As the electron transport system continues to function, a high concentration of protons builds up in the intermembrane space. This creates a proton gradient across the inner mitochondrial membrane. Such a gradient is an enormous energy source, like water stored behind a dam. The mitochondria make use of the potential energy of the gradient to synthesize ATP energy. ATP synthase harvests the energy of this gradient by making ATP. Protons pass through the F0 channel back into the matrix. This causes F1 to become an active enzyme that catalyzes the phosphorylation of ADP to produce ATP. In this way, the energy of the proton reservoir is harvested to make ATP. If we take a look at the summary of the energy yield from one glucose molecule, and glucose has 6 carbons. Its molecular formula is C6H12O6. We'll start by looking at the oxygen-independent metabolic pathway, known as glycolysis. And in glycolysis, we produce 2 ATPs from substrate-level phosphorylation, and we also produced 2 NADHs in glycolysis. And normally we think of an NADH being the equivalent of 3 ATPs, as is shown down here. So here's the conversion factor down here in the citric acid cycle. But that NADH that's produced in glycolysis, we call this cytoplasmic NADH, and it's only got a value of 2 ATPs. So 2 times 2 gives us a total of 4 ATPs. Therefore, the net yield of ATP from glycolysis is 6 ATPs from that glucose molecule. Then, of course, in glycolysis, not only do we produce 2 ATPs and 2 NADHs, we also produce two pyruvate molecules, and we have to convert those into acetyl coenzyme A so that they can enter the citric acid cycle. And in the process of taking those pyruvates and converting them into acetyl coenzyme A, we produce 2 NADHs, and each one of those is worth 3 ATPs. Therefore, 2 times 3 gives us a total of 6 ATPs. And since we split that glucose molecule in half, we end up going from a 6-carbon molecule to a 3-carbon molecule, pyruvate. And that is why we do 2 turns of the citric acid cycle, because we go from a 6-carbon molecule to a 3-carbon molecule, our pyruvate. So we take our 2 acetyl coenzyme A molecules, they enter the citric acid cycle, and in the citric acid cycle we produce 1 GTP, but since it's 2 turns, that's 2 GTPs. Each GTP is worth 1 ATP, which gives us a total of 2. Then again, we produce 3 NADHs, but of course it's 2 turns, so 2 times 3 is 6. Each one of those is worth 3 ATPs, which gives us a total of 18 ATPs. And then we produce also 1 FADH2, but of course it's 2 turns, so it's 2 FADH2s overall. And each one of those is worth 2 ATPs, which gives us a total of 4. And so when we tally up the net yield of ATP from the conversion of pyruvate into acetyl coenzyme A in the citric acid cycle, we get 30. And therefore the total ATP that's produced via glycolysis, the conversion of pyruvate to acetyl coenzyme A, and then the citric acid cycle is 36 ATPs. That's what we get as an energy yield from 1 glucose molecule.